So yesterday we learnt in Bangla good evening, Shubho Shondha. It's not evening yet, starts at 6 p.m. So it's good afternoon. Good afternoon in Bangla is Shubho Apuranno. So Shubho Apuranno to everyone. Welcome to this session on early life childhood diseases. I am going to chair this session along with my co-chair, Archita. We're going to get started because there is no coffee. It's not my fault, it's Dr. Partha Majumdar's fault. Do not associate this face with the lack of coffee. Our first speaker is Dr. Arindam Maitra, and he is coming here from Kalyane. Please, Arindam.
Thank you, Sajuti. Continuing the Bangla lessons, Dhonnobad, which means thank you essentially. Two countries speak the same language. And thank you, Pachoda, for cancelling the break. I am getting sleepy, so I would like to move on. I am Arindam, and I come from National Institute of Biomedical Genomics. As my colleague earlier pointed out, it's the only institute in India which is dedicated to the cause of genomics of human health and disease, whose founder director is Professor Partha Majumdar. Today I will be talking to you about a different aspect in this session, which is on our maternal and child health, some of our maternal and child health research. So before that, let me shift gears and explain to you, talk to you a little bit about why studying maternal and child health and early childhood disease is important, in particular focusing on pregnancy outcomes, particularly a particular trait of the pregnancy outcome. It's called preterm birth is where we are interested in and working on. Preterm birth is defined as birth before 37 weeks of gestation. And for various reasons, it has long-term implications on the maternal health, as well as there is a big impact on child health. Babies born preterm have very high rates of neonatal mortality, delayed development during early childhood, and even increased risk of late onset adult diseases. So, in preterm birth is a leading cause of neonatal mortality almost, and particularly working in Southeast Asia and India, it's a very important place to study preterm birth because India and South Asia has the highest rates of preterm birth and contributes the highest number of babies born preterm to the globe. But however, studying pregnancy is not easy. Pregnancy is a complex, dynamic physiological changes, and there is a lot of heterogeneity in the underlying mechanisms of pregnancy outcomes like preterm birth. So, but if I have been able to convince you that studying pregnancy outcomes like preterm birth is very strategic for public health perspective, because they result, if we can control these outcomes, we can control and increase the birth of healthy babies, which will lead to a healthy world in future. So I work on a very large cohort in a program. It's called the Garbhini cohort. It's a short acronym for a larger name. This cohort is basically established in May 2015 in a northern part of India called it's a place called Gurugram or Gurgaon. It is very close to Delhi. And the idea of this cohort was to study multidimensional correlates of preterm birth in India. And there are multiple organizations involved. I work on the genomics, epigenomics, and transcriptomics of preterm birth in this particular cohort. It started with recruiting 8,000 pregnant women. For every trimester, we collected different biospecimens, health records, and ultrasound data. So it's a very well annotated cohort. But now we have not only covered 8,000 women, but we have now actually moved in and reached about 12,000 women in the cohort. So in the genomic arm, we wanted to identify what are the maternal genomic underpinnings of preterm birth in India. And to do this, we conducted to our knowledge, the first GWAS on preterm birth in South Asia, which was based on 6,211 women at that time when we did the GWAS. We use the Infinium global screening array with multi-disease drop-in panel, which is about 700,000 SNP markers. We did a full cohort analysis where we had 521 preterm delivering women and 4,161 term delivering women after various kind of QA/QC measures where we had to filter off a few women. So this was the GWAS, and to cut a long story short, we actually, when we looked into our GWAS data, we found that we identified one SNP which was significantly associated with preterm birth at the level of genome-wide significance, which is p-value less than 10 to the power minus 8. This SNP, which is in short I will call it RS735, in addition to this SNP, we also identified about 39 other SNPs which had a suggestive level of significance. Now, when we look back, and there is a sub-phenotype of preterm birth, which is known as early spontaneous preterm birth, where the children are born neonates are born less than 33 weeks of gestation. We found that about 50% of the SNP markers are actually associated with this early spontaneous preterm birth. Coming back to the genome-wide significant SNP, we found that in literature it had some evidence that it controls, it's a cis-sequential of a gene called NRF2, and the risk allele, which is the G allele, actually is associated with, has a higher frequency in the preterm delivering women, is significantly associated with a lowered gestational age at delivery. And this NRF2 expression is enhanced by the G allele, and that results in a downstream effect of increasing the VGFA-VGFR signaling, and that leads to arterial stiffness and ultimately results in defective development of maternal placental bed and resulting in preterm delivery. Now, when this arterial stiffness increases, there were clinical features of this, and that can be measured by arterial pulsability index. We found that this G allele is actually associated with an increased arterial pulsability index in the women that we genotyped. And we looked at the published single-cell, early trimester single-cell transcriptomic data. We found that this particular gene, NR2F2, is mostly expressed in the trophoblast and some of the perivascular cells. So ultimately what we hypothesize is that this particular SNP or RS735G allele may result in preterm delivery by controlling NR2F2 expression, which results in poor maternal placental development. There's a large GWAS conducted on European women on preterm birth based on the 23andMe cohort. And we looked at that was published earlier in New England Journal of Medicine. We looked at that data, and with some difficulty of accessing it, we finally managed to get access to some of the data. And we found that there are about 190 SNPs which were significant in this GWAS, are also significant to certain varying levels of significance in our cohort as well. We found that the topmost significant SNP was RS881, and we found that this particular SNP is kind of transethnic in nature because it is polymorphic to a varying extent across all non-European populations globally. Since this was an imputed SNP, and we wanted to genotype it in our cohort and see and we found that it actually concords very well with imputed genotype data. And so what we found is that this particular SNP, the risk allele is A, and this SNP is a cis-sequential of a gene called AKIP1. The A allele increases the expression of AKIP1, and that is known to result in increase of NF-kappa beta expression, leading to a pro-inflammatory state, leading ultimately to myometrial contraction, fetal membrane rupture, and cervical ripening, which might result in preterm delivery. Now, we went back and found that there were two interesting things. We found that this risk allele is associated with an increase in pulse pressure, and also found that this allele is also associated with a decrease in cervical length. So both the inflammatory state and cervical ripening, bilateral contraction and cervical ripening are indicated by the association with these clinical features in our cohort. We also found in the published single-cell data that this particular gene is expressed mostly in the immune cells, particularly the Hubbar cells and the macrophages. So we hypothesized that this transethnic SNP may result in preterm delivery by increasing the pro-inflammatory state and cervical ripening. So we also did bulk transcriptome sequencing of placenta of preterm delivering women and term delivering women in our cohort, and we found, to cut a long story short, we identified various upregulated and downregulated genes. In particular, today I am focusing on these two genes which we identified in GWAS. We found that there is a increase in expression of both these genes in the preterm delivering women's placenta compared to those delivering term. Now, we used again, went back to the Ventotomo data, which is published on placental single-cell sequencing on early trimester pregnancies, and we used CyberCert X to define signatures of various cell types for deconvoluting our bulk transcriptomic data. We then looked into our data and found, based on these data sets, that there is an increase in syncytial trophoblasts in the preterm delivering women and a decrease in the villus cytotrophoblast in the preterm delivering women. Now, it has been known that there is this might lead, what it suggests is that in preterm delivering women, there is a premature accelerated differentiation of VCTs to SCTs, which might be associated with elevated levels of human chorionic gonadotrophin and inhibin A, which leads to placental dysfunction. In the deconvoluted data set, we also found that there is an increase in macrophage proportions and fibroblast proportions in the preterm delivering women, although they were not significant. But there was a trend, and which suggests an elevated inflammation and a pro-inflammatory responses generated by the macrophages and mediated by fibroblasts through IL-33-ST2 expression, which can result in preterm delivery. So we have also right now conducting a single-cell sequencing study on the placenta of these women in our cohort who deliver at various gestational ages at delivery. This is an ongoing work, and we have just finished doing the placental analysis for 4 term delivering women, and when we went into the data, we have now just identified all the various cell types and cell states in the term delivering placenta. And to cut a long story short, we looked into this. One gene was NRF2F2. We found that similar cell clusters as in the Ventotomo, the early trimester pregnancies, also express NR2F2 in the term placenta that we analyzed, although at lower levels of expression. We hypothesized that NR2F2 actually regulates transcription factor TFA2P2A, which actually might be crucial for terminal differentiation of VCTs to SCTs, and that's how we can tie up our GWAS data set, the bulk transcriptome data, and the single transcriptome results. We also looked at AKIP1, and we found that very few cells in our term placenta actually express AKIP1, which is not surprising because these are term. And we think that AKIP1 resulting elevated mostly in the Hubbar cells in early trimesters actually would lead to expression of pro-inflammatory cytokines. And our data suggests that expression is reduced at later gestational ages. So in our term placenta data, we have now looked at the trophoblasts because we think that the NR2F2 implicates trophoblast differentiation. So we found that the proliferative VCTs actually evolved into non-proliferative VCTs, and then they differentiate into SCTs and EVTs. That is very apparent in our term placenta data set that we have generated. And we also found that there are higher elevated genes which are expressed as the S and G2M phases in the proliferative VCTs, whereas we find that there is an increase in invasive gene signatures in the SCTs and subpopulations of EVTs. We then also now have looked into various immune cells we have characterized, and we have also identified various rare myeloid and lymphoid cells also in this population. This work is ongoing, and actually we'll be able to get better insights after we complete analysis of the preterm placenta at various gestational ages at delivery. So with this, we would like to summarize that we found essentially our results actually imply that there are two important aspects of preterm delivery. One is coming from the placenta, the poor formation of the placental bed or placental dysfunction, and the other is from inflammation. So our results point to these two important factors which contribute to preterm delivery. In our cohort, and the inflammation might be a trans-ethnic phenomenon anywhere in the world that can cause preterm delivery. So I will stop here and acknowledge many people with whom I have worked, and we are working to get this, particularly my student Aisha, who spearheads the GWAS. Piali and Supratim are here, who are spearheading the placental analysis of single cell and bulk transcriptome. Professor Majumdar and Shinjini Bhatnagar, who spearheads the cohort, and Pallavi, my colleague in THSTI, who actually provided the placenta and collaborates on the placental analysis. Funders are of course acknowledged, and overall the Garbhini cohort, the participants, and the enormous team of data collection clinicians, everybody who puts together substantial annotation for us to do such wonderful research. Thank you very much, and I would like to take any questions if there are any.
I already see a question from Archita. If you have questions, please walk up to the microphone.
Because you say, am I on? Okay, great talk, Arindam. I was wondering, because you talked about inflammation, so the differences that you find in your Garbhini cohort, which is a phenomenal cohort, are there any external factors relating to what mother took during pregnancy, like antibiotics or gestational diabetes? Anything like that came into picture for preterm birth?
It's a very good question, actually. So for our genomic study as well as for the placenta, we have for the time excluded all women who had been taking any medication and who had other comorbidities or any other physiological problems, so that we can reduce the confounders as much. So, but in the overall cohort, we are also addressing these questions, and hopefully sometime that story will unfold.
Thank you. I have a question.
Sure.
So I think this study is so relevant to our part of the world where preterm birth is so common. So what is kind of the incidence? Do we have any prevalence data of preterm birth across India, and are there regions where preterm birth is more common? A, and B is, I might have missed it, but what was your sample size for preterm placental samples, and did you have any challenges collecting those samples, because usually they are not vaginal delivery?
So, so all this placenta we took were, they were all vaginal deliveries, so we excluded all the other kinds. Again, there were many challenges. This data that I showed on term placenta was on about 5 placenta samples that we did. There are many more that we have collected and analyzing, but for this we took 5 which are absolutely fitting all inclusion criteria as stringent as possible. And as far as the prevalence goes, the literature says, the data says that there is about 10% deliveries are preterm, but in our cohort we have found that there is a slightly more, so around 12.5% of the deliveries we found were preterm, spontaneous preterm deliveries. Thank you.
Great talk, Arindam. Thank you. And the next speaker is Gunjan Dixit. She's a bioinformatics expert and currently pursuing her postdoctoral studies in Australia in Peter Mac Center under Professor Alicia Oshlock. And she'll be talking about the phenomenal work she's doing in pediatric atlasing in the airway epithelium. Gunjan, welcome on board.
Good evening, everyone, and thanks, Archita, for such kind introduction. I have 10 minutes, and I'll try to be really quick. So today I'll be talking about building the Pediatric Airway Atlas, one tissue at a time. So jumping straight to the main aim of this project, so we aim to recruit healthy participants spanning early age childhood to adolescent and using 10x Flex to construct a comprehensive cell atlas from the 5 different tissues. So nasal brushings, tonsils, and adenoids representing the upper airway, and bronchial brushings and bronchiolar lavage representing the lower airway. And we want to build this atlas to advance our understanding of the immune and epithelial development in childhood. So you might have come across this golden circle, which is usually seen with business models, but I try to associate it with research projects as well. So I briefly described the main aim of the project, and I'll come back to that later. But coming to how we're going to do what we're going to do, and that brings me to the question of what is 10x Flex. So without going into too much detail, you can actually scan the QR code or check the 10x website to get more details. But to give you a brief overview of what 10x Flex is, it's a probe-based method which allows you to fix your samples in paraformaldehyde, and then you can store them up to 6 months. And then you can hybridize them using a probe set and pull them once you have enough samples. So this is a huge project and divided into 3 stages. So MCRI does the sample collection and pooling, etc. Then the VHI team does the library construction and the sequencing. And all the computational aspect and data analysis is taken care by the PeterMac team. And now coming to what are the actual results so far. So giving you a snapshot of the data distribution first. So we have 5 tissue types and 2 batches of each of those tissues. And we have 16 samples in each batch except for the BAL. And the reason is that collecting bronchiolar lavage samples is quite an invasive process. And usually a child needs to be under anesthesia to collect those samples, and that is not something a person would go regularly for. So we could only manage to get 8 samples for the bronchiolar lavage. And for the nasal brushings, we had an extra or spare spare group giving us technical replicates an additional 9 samples. Next, looking at the multi-tissue data distribution across the participants. So we had at least 11 participants who had given the data or the samples for 4 different tissue types. Then we had 14, 16, and 17 respectively for 3, 2, or 1 tissues. Next, looking at the age and sex distribution across the tissue samples. So in this box plot, I have tissues on the x-axis and age on the y-axis, and each dot represents a sample. So we could manage to get samples fairly distributed across the 3 target age groups that we were looking for. And similarly for the sex distribution, although the number of male samples are a bit higher than the female samples, but it is not extremely biased towards one category. So next, looking at the number of cells or non-empty droplets that we achieved for each of the cell type, we got the highest number for tonsils and adenoids, the solid tissues, followed by the nasal brushings, bronchial brushings, and the bronchiolar lavage. And that gave us a total of approximately 796,000 cells, which I believe is quite a high number and definitely worth building the atlas for. So I'll be now talking about tonsils, not because it gave us the highest number of cells, but also this was the first batch that I started working with. Not going into the details of the method section, but we followed the standard protocol of the QC, identifying the empty droplets, looking for doublets, doing the cell clustering, and cell type annotation. So since there is already an adult tonsil atlas available on Azimuth, we used that as a reference to map our data. And the results we got were quite consistent with the adult atlas, and the major cell types were the naive B cells, germinal center B cells, CD4T, memory B, and cycling B cells. I just have enough time to cover one result in detail. So we wanted to look the age-related shifts in the tonsil cell atlas. So this is for the adult atlas data. And in this bar plot, I have age on the x-axis and I have proportion of cell types on the y-axis. So we can see there are significant changes in some of those cell types as we go in the age. So especially for the CD4 T cells, cycling B cells, germinal center B cells, and memory B cells, the numbers are drastically changing towards the end. And even if you look at the 5-year age or the 26-year age in the center, these numbers are significantly changing. So this sort of highlights the need for the pediatric cell atlas and for us to recognize these progress and to fill these gaps. So next, I tried to plot the same bar plot with our data. So here I have samples on x-axis arranged in increasing order of age. So this one being the youngest and towards the left being the oldest. And we see these cell type proportions changing as the age increases. So to further explore this relationship, we use some statistical testing, and these are just very preliminary results that we plotted, and we need to put in more factors in consideration before jumping into conclusions, but I just wanted to show that there is definitely some sort of pattern that we can observe. So for example, for the CD4 T cells, using continuous age as a factor, we put the regression model, and we can see an increasing proportion as we go towards the increasing age. And for the germinal center B cells, the pattern is sort of decreasing, but not very sure. And if you group these into 3 different categories of the kids, preschool and adolescent, the difference is much clearer, and you can see there's definitely a pattern. So again, as I mentioned, these are just preliminary results, and we need to further validate what we're doing and consider more factors when we're setting these models. So that brings me to the last slide. So to summarize, we've defined the immune cell profile of the pediatric atlas of tonsils at the moment, and George has defined for the nasal brushings. And we have more tissues to look at, and we're aiming to build this atlas with approximately 800,000 or more cells. Next, we're using this probe-based 10x flex method, and we're learning new things about it as we go. So that is something that we're figuring out as well. And there are age-related shifts that we have been seeing in cell type proportions for tonsils so far. And for the significance, I'd like to highlight this quote by D.N. Taylor that kids are not mini adults. They have their own set of diseases, their own set of symptoms, and hence they need their own atlas. And especially because most of the development occurs in the earlier stages of life, hence it is quite crucial to understand how these changes are occurring to find better ways to treat those diseases. Next steps, so there are always some ifs, buts, and maybes associated with a project. So currently, as I mentioned, we're working with more tissues. But at the same time, we're finding out answers to questions like, should we treat the 10x flex similar to the 3' sequencing? And also, do we need additional QC steps to address the ambient RNA or the background? Because we found a lot of ambient RNA in the flex data that we obtained. I've highlighted 2 preprint and a recently published paper of our lab to give you a glimpse of what we've been working on. And finally, I'd like to acknowledge CZI for the funding, especially Mel and Neeland from the MCRI team for their biological insights, and Alicia, Yovana, George, and Calandra for all the effort that they've been putting behind this project. Thank you. I'm happy to take questions.
Thank you so much, Gunjan. Anybody has questions here? That was a great talk, Gunjan, and I think the fact that children are not adult, like tiny adults, it's very important to highlight. And thank you so much. Yeah, in the respect of time, we'll move forward.
Yep.
I'll
Thank
ask a
you.
question.
Sorry.
Okay.
I have a question, and
Yep.
it's on your last slide.
Okay.
So I learned about 10x Flex for the first time yesterday, and we had a short conversation too. And from your last slide, it seemed like you have some feelings about 10x Flex, right? So would you comment a little bit on the pros and cons, or your experiences so far as you're analyzing the data?
Yeah, so there are certainly several advantages that come with Flex. So you can store your samples. So especially when you're working with clinical samples or collecting healthy samples, you don't know when the next sample would come in. So one of the advantages that you can store the samples, and when you have enough samples, you can process them together, and it also reduces the cost per sample cost as well. Another advantage of Flex is that you can ship and store and transport your data, and doesn't affect, reduces the batch effects. So it has several advantages in those terms. But the disadvantages at this moment would be that it's quite new for us as we're analyzing it for the first time. So there's not enough insights available of what to look at. So when we looked at the Flex data for the first time, we saw a lot of mixing in the UMAP, and that's when we noticed that we have a lot of ambient RNA. And the normal methods that we were using to get rid of those background, like SOUPEX and stuff like that, they were not working very well with the Flex data. So that made us realize that maybe we need additional QC steps to figure that out. So yeah.
Dhanyabad, Gunjan.
Thank you.